This is Jessie Hunt, Chapter 3 Outline, Segment 1, pages 59 to 65. The Laughing Brain. Deirdre experiences seizures which impair her daily function. She decided to do a surgery where doctors implant tiny wires to pinpoint the electrical problems in Deirdre's brain. They can then decide whether or not to remove the part of the brain that is affected. The doctors stimulated an area in her brain which made her laugh and find normal things amusing. Even functions such as a sense of humor arise from the mechanisms of the brain. Neurophysiology is the study of electrical and chemical processes in a neuron. Electrical signals are the vocabulary of the nervous system. All living cells possess an electrical charge. They are more negative on the inside than the outside. The inside negativity of living cells comes from the proteins inside them, which are most often negatively charged. Neurons evolved to exploit this charge difference and to transmit electrical signals. The electrical signals are much the same from jellyfish to humans. A balance of electrochemical forces produces the resting membrane potential of neurons. An ion is an electrically charged molecule. Anions, the majority of ions in the neuron, especially large proteins which cannot exit the cell, are negatively charged molecules. Cations are positively charged ions. All of these ions are dissolved in an intracellular fluid. The cell membrane separates the intracellular fluid from the extracellular fluid. The cell membrane is made of a lipid bilayer. There are more negatively charged ions than inside the neuron relative to the outside of the neuron. We measure the difference in charge across a membrane by inserting a microelectrode and using a voltmeter, as shown in Figure 3.1. The resting membrane potential of a neuron is about -50 to -80 millivolts. The negative sign indicates the negative polarity. An ion channel is a tube-like pore which allows ions of a specific type to pass through the membrane. Many channels are open constantly, such as the channels which selectively allow the transport of potassium ions. Selective permeability is the property of a membrane being selectively permeable for certain substances, such as potassium ions. The resting membrane potential of a neuron is controlled by opposing forces pushing potassium in and out of the cell. These forces are 1. Diffusion, the tendency of molecules to move down their concentration gradient from areas of high concentration to areas of low concentration, and 2. The sodium-potassium pump, which pumps 3 sodium out for every 2 potassium in. This action consumes energy in the form of ATP, as shown in Figure 3.3a. Pushing out of the sodium-potassium pump causes the buildup of potassium in the cell, but the membrane is permeable for potassium, so the ions will flow down their concentration gradient out of the cell, causing there to be a net of negative charges inside the cell. As negative charges inside the cell build up, it causes electrostatic pressure to pull potassium back inside the cell. These two forces, the concentration of gradient pushing potassium out and the electrostatic pressure pulling potassium in, causes equilibrium when the inward movement of ions equals the outward movement of ions. The Nernst equation is the mathematical function predicting the equilibrium potential of a resting membrane. The equation can be seen on page 65. There is a discrepancy between what the Nernst equation predicts the resting membrane potential to be and what it actually is. This is because the Nernst equation makes assumptions. It assumes that the membrane is permeable only to potassium. It is, in fact, somewhat permeable to other ions too. The Goldman equation takes into account the intracellular and extracellular concentrations of potassium and sodium and other ions, and it takes into account the degree of permeability of each ion. This equation has a much more accurate prediction of the resting membrane potential. Figure 3.4 shows the concentrations of various ions inside and outside of the cell. Take note of the high intracellular concentration of potassium and the high extracellular concentration of sodium. These two concentrations are the result of the action of the sodium-potassium pump. The low intracellular concentration of calcium. This is because of another ion pump which ejects calcium from the neuron and specialized proteins which store calcium for use in intracellular signaling. The resting potential of a neuron is the same as the baseline level of polarization in every cell. However, neurons frequently go through a brief but radical change in polarization.